Maybe we'll give folks like 1 more minute to log on. Is that okay? Okay, I think I probably got started. Welcome everyone to the last Geriatric Medicine Grand Rounds of 2022. We're getting toward the end of the year. So welcome all of you, and I hope you all can have a little bit of time to rest around the holidays. We have a really great speaker today, Dr. Elizabeth Rhea. Many of you know, you see her on our conferences every week, and many of you work with her. She is a research assistant professor based at the Seattle VA, and has done quite a bit of work advancing our knowledge in the blood-brain barrier in particular, has a specific interest in how peptides, regulatory peptides cross the blood-brain barrier. And you might remember some of her work that she has done during the pandemic, helping us understand how the virus that causes COVID-19 can cross the blood-brain barrier and do what it does. So I am really excited that you're going to talk to us about some of your work that you do with the Alzheimer's disease research center, and this work that you're leading related to insulin and Alzheimer's disease and its interaction with the blood-brain barrier and the brain. So thank you so much. This is like really cutting edge work, and we're really, really lucky that we get to hear all of this before it hits prime time, because this is where knowledge is evolving, and you're at the cutting edge. So thanks for.
Thank you, Kate, for that introduction, and thank you all for being here today. I hope everyone's surviving the flu and RSV and eternal COVID season, and at least managing that. I know a lot of people have been affected by it this season, so I just want to thank you for being here. And like Kate said, I hope to convince you all about the importance of brain insulin resistance, specifically in Alzheimer's disease, and present why this is a clinical problem, but use a lot of my basic science research to help push this field forward to understand what's actually going on in the brains of these Alzheimer's disease patients. So the goal is basically to update everyone in the division on what I've been doing over the last couple of years. I think it's been since 2020 that I last presented, and while I won't be speaking to any of the COVID-related work today, I will speak about how our group has kind of barrier transport and brain insulin signaling. So of course, I'll start off defining what CNS insulin resistance is and what the role of insulin in the brain is, for those of you that might not be too aware in this field. I'll briefly introduce the blood-brain barrier, because there are quite a few other groups in this division that work on the blood-brain barrier. I'll present what the characteristics of insulin blood-brain barrier transport are. And then I'll get into my research regarding the CNS regulation of insulin blood-brain barrier transport, which was actually just submitted earlier this month. And then I'll speak to ways that we're working to try to reverse CNS insulin resistance. And the first part is through exercise, which was an exciting project a couple years ago. And the second is through this more novel approach, including the use of incretin receptor agonists, which many of you may be prescribing in the clinic currently, as it's the primary prescription for type 2 diabetes. And feel free to ask questions throughout, either raise your hand or just speak up or put it in the chat and I'll try to monitor. So I wanted to present this first kind of global overview of insulin in the body and where the insulin receptor is expressed. And so looking at RNA-seq data, you can see that the insulin receptor is expressed pretty much in every tissue throughout the body. Every cell type, I haven't come across a cell type that doesn't express it. The pancreas obviously has the most insulin receptor and a vascular network. The aorta has the next most abundant insulin receptor. But I want to point out the brain isn't far behind. And in fact, it's greater than levels in the liver, adipose tissue, and skeletal muscle, which are some of the most important metabolic regulators in the body. So the brain should not be left out when thinking about the role of insulin in the brain. And here's a recent review that I collaborated with Bill Banks and Jacob Raver, and we kind of outlined some of the important roles of insulin throughout the body. Of course, all of the peripheral roles, and even in the brain, insulin has a peripheral role regulating hepatic gluconeogenesis, regulating blood flow and appetite. But it also has this important role in the brain, acting in learning and memory, reward. It interacts with amyloid pathology. And so it really has this two-function role in the brain. And so this role in learning and memory is what I'll focus on today. So how is CNS insulin resistance defined? Well, the classic definition of insulin resistance is technically the absent or inadequate response to insulin. And so that can be applied to the brain. But as I'll get into in the next couple of slides, the brain has an added step in this CNS insulin resistance. And so what's the prevalence of insulin resistance in the US population? Well, for those over 50, approximately 40% have some form of peripheral insulin resistance. Some studies suggest, and some investigators propose, that approximately 90% of people with Alzheimer's disease actually have CNS insulin resistance. And I want to make this point because a lot of people think of peripheral insulin resistance driving Alzheimer's disease, and type 2 diabetes is a risk factor for Alzheimer's disease. But brain insulin resistance can exist without peripheral insulin resistance. So they can be independent from one another. And that's this point below, in which just 35% have both Alzheimer's disease and peripheral insulin resistance. So how do we know that there's even brain insulin resistance in Alzheimer's disease? What is driving this 90% number? Well, first, it's been shown that patients with Alzheimer's disease have decreased levels of brain insulin compared to age-matched control brains. So their levels of insulin in the brain are reduced. The amount of that insulin receptor that I mentioned earlier is decreased with age. Insulin signaling is impaired, meaning the subsequent downstream signaling processes following receptor activation. And it's also been shown that insulin, Dan Kraft, who was here about a decade ago now, and she showed that using a hyperinsulinemic euglycemic clamp, that individuals had improved memory. And this is important because it shows that it wasn't the glucose driving that improved memory. It was truly related to the insulin. She's further gone on to show, and others have shown and repeated this numerous times, that intranasal delivery of insulin can improve memory. And so that's just another point of evidence for CNS insulin resistance. And lastly, I want to bring up some animal studies in which the level of the brain insulin receptor was manipulated, whether it's overexpressed or knocked down, and they saw what they would expect. With overexpression, they saw improved cognition. With knocking it down, they saw impaired cognition in these rodents. So it really drives this insulin receptor signaling process and its role in cognition. And so, as I mentioned, the brain is at increased level of regulation when it comes to brain insulin resistance because it does not make its own insulin. Insulin must cross the blood-brain barrier from the blood and get into the brain to have an effect within the brain. And so if this transport system is altered, then that could potentially lead to brain insulin resistance or impaired insulin signaling. Another step is the insulin degrading enzyme. If this enzyme that actually degrades insulin is overactive, then that's going to reduce the availability of insulin within the brain. And then these last few points are all what's important in peripheral insulin resistance, and that insulin receptor level, the binding affinity for insulin to be able to bind the insulin receptor, and then activation of it through phosphorylation signaling events can also be points of CNS insulin resistance. So getting back to the levels of the insulin receptor and where it's expressed in the brain, the first figure I showed you was throughout the body, and now we're going to hone in on the brain, next step level. And so this is all RNA-seq database, so it's mRNA transcript levels of the insulin receptor. And if we look at the cells that are involved in the blood-brain barrier, endothelial cells, pericytes, neurons, and astrocytes, we can kind of see the distribution of this receptor throughout these cell types. And I want to point out that the distribution is pretty similar between human and mouse. And so it suggests that expression of this protein or expression of this gene is pretty conserved across species. And what I also want to point out is that the endothelial cells have the most abundant levels of this insulin receptor transcript, suggesting that it is an important signaling protein in these cells, which I'll get to in some of my data slides. This graph on the right now takes just the vascular cells, so just those endothelial cells, and now looks at it across the vascular beds within the brain. And so we've got capillaries, we've got veins, we've got arteries. And so it's comparing the level of the insulin receptor in these different vascular beds. But what I want to point out is while the majority is in the capillary bed where nutrient exchange occurs and where the signal is most often relayed to the brain, Alzheimer's disease patients have reduced levels in pretty much all of these vascular beds. So it suggests some sort of dysregulation going on. So what is this blood-brain barrier? The blood-brain barrier is a unique structure within the brain, and it's formed by these endothelial cells that are connected by tight junctions. These endothelial cells are surrounded by basement membranes and pericytes, which help support the tightness of these endothelial cells, as well as the function, so transporters at this system. And then you have astrocytic infeet that surround these endothelial cells. And I just learned in a talk a couple of days ago that actually every single astrocyte in the brain has at least one infoot that touches an endothelial cell, which is just kind of mind-blowing to think about. If you think about how many astrocytes there are, that one of these is at least touching the endothelial cell, and thus there's this strong communication going on between the astrocytes and the brain endothelial cells. So this blood-brain barrier is what's important in the brain and helps regulate what actually enters the brain, including insulin. So what do we know about these this transporter characteristics for insulin? Well, we know it doesn't really transport other hormones, including leptin and amylin, except for maybe small amounts of IGF-1, but the complete selectivity is unknown. You know, we obviously can't test every single hormone and whether that interferes with the ability for insulin to get into the brain. But of many of the ones that have been tested, insulin still seems to be pretty selective. Insulin transport still seems to be pretty selective. We know it's saturable, meaning at higher blood levels, we don't see a greater amount entering the brain. And we know it doesn't depend on the endothelial insulin receptor. So I've talked a lot about the insulin receptor, especially in endothelial cells, which maybe you thought that was going to be what was actually getting insulin into the brain. But what we found a few years ago was that if we either knock out this protein specifically in endothelial cells in these mice, or if we inhibit this insulin receptor using a pharmacological inhibitor that has a high affinity for the insulin receptor, and we look at insulin transport into the brain, we see that the slopes are not different. So in either graph, insulin's getting into the brain at the same rate, the slope hasn't changed, which is indicative of the rate. But what you can appreciate is that these colored lines are much lower than the solid wild type vehicle black lines. And that's due to the ability for insulin to bind this vascular structure. And so here we've eliminated one of the major binding sites for insulin in the vascular endothelium. And we in fact see this decrease in the ability for insulin to bind. So this supports our models. It suggests that our insulin receptor really was lost in these vascular beds or inhibited, but insulin transport still prevailed. We also know that the rate of insulin transport is variable amongst all the brain regions. So in many brain region animal studies, it's been shown that the olfactory bulb has the fastest rate of insulin transport. And then there are other regions like the midbrain thalamus and occipital cortex that have very little. We know what the average rate of insulin transport into the brain is. So if we have a manipulator that changes this rate, we can determine whether it's increasing or decreasing it. And this is similar to other hormonal peptides that can regulate appetite. And it's also affected by many different factors. So I don't expect you to read through this list of all the factors known that we were able to pull from the literature that can regulate insulin transport. But this work was this was a recent review that Bill and I put together with an undergraduate in the lab, Cassidy Noonan. And she essentially combed through the entire literature, all the reviews that have been published, all the primary papers, and looked at what modulators of insulin transport have been investigated. She found some increased, some did not change insulin transport, and some decreased insulin transport. And so really, these are mostly peripheral factors. So obesity or a short-term fasting can actually decrease the interactions of insulin in the blood-brain barrier. These are just a few examples of what's on the list. Whereas like diabetes or long-term starvation can actually increase these interactions. And this list is ever growing. So what I'll present today are two things that I've recently added to this list, in which we found CNS insulin signaling can actually affect or decrease the ability for insulin to bind the blood-brain barrier, interact with the blood-brain barrier. And we find exercise actually can increase these interactions. And I'll get into the detailed data later. So the first question I want to jump into was that point on the right, and that's whether brain insulin signaling. Mentioned that a lot of these factors are peripherally mediated or peripherally regulated or serum substrates. And so we wanted to go into the brain. Can the brain control how much insulin it needs at any given time, which made sense because of this feedback mechanism. If the brain needs insulin, it needs a way to signal for that. So this work was started quite a few years ago by a very talented undergraduate in the lab that I had for a fortunate amount of time, 3 years, Sarah Pemberton. She's now gone on to work with another group at Seattle Children's after graduating last year. A new technician in the lab, Peter Thomas, and it was wrapped up this summer by a talented medical student through the MSTAR program that May Reed manages, Van Nguyen. And so together, we all investigated different forms of brain manipulation to determine whether insulin blood to brain transport was affected. So we either gave intranasal insulin, which is what's being used in the clinic. We wanted to make sure the endogenous transport system wasn't being affected by all of these clinical endeavors to try to treat brain insulin resistance. And then we also looked at direct delivery of insulin to the brain, or direct delivery of this inhibitor for the receptor to the brain, S961. And after a period of time, we then used our radioactively labeled insulin to look at the blood to brain transport across the blood-brain barrier. And so the direct delivery of our substrates to the brain involved intracerebroventricular injections, which is an injection straight into the lateral ventricle. And you can see the coordinates here with the related to the bregma of a mouse brain. And so we're truly injecting it straight into this ventricle. So first we wanted to see when we inject throughout the brain. We want to make sure we're affecting the entire brain because we're looking at whole brain blood-brain barrier transport. So when we give intranasal insulin, which I've presented before, so I'm not going to spend a lot of time on, we see in all mouse models that we've really investigated that it distributes throughout the brain. It appears within 5 minutes and it kind of peaks around 30 minutes. So that's why we stuck with this 30-minute time point for intranasal delivery. For ICV delivery, we also see that insulin can distribute throughout the brain, and then our inhibitor can also distribute throughout the brain in this time frame of, we stuck with 10 minutes for this delivery point. So we know our substrates are getting throughout the brain. So now we want to look at that blood-brain barrier transport. So now we're giving intranasal insulin, we're waiting 30 minutes, and then we do our radioactive pharmacokinetic study. And whether we give vehicle or insulin, you can't really tell a difference between the two lines. The slope is not different. The appearance on the graph is not different. And this indicates that when we give intranasal insulin, we're not affecting insulin transport into the brain, which is good for those looking at it as a clinical therapeutic. If we give it directly into the brain, which delivers a lot more insulin to the brain, so that's the difference between intranasal and our ICV delivery, is we're getting a lot more when we deliver it ICV because everything is going straight to the brain. We actually start to see a flattening of the insulin line, and this indicates that insulin transport into the brain is being slowed when insulin is in abundance in the brain. And we see this both in the whole brain and the olfactory bulb. So this piece of data told us that if we have too much insulin in the brain can say, hey, we don't need any more insulin, please slow down the transport rate from the blood. We're good on signaling right now. Now we wanted to test the alternative. If the insulin receptor is inhibited or impaired as it is in brain insulin resistance, does this affect insulin blood-brain barrier transport? And what we found was that it actually does, at least in females. So if we block the ability for insulin to signal within the brain, we slow this transport rate. And this was really important to show because we've been wanting to link blood-brain barrier transport with brain insulin resistance for years now, and this piece of evidence finally helps support this, that if the brain is in this insulin resistant state, we also get impaired blood-brain barrier transport. And so if we can reverse one or the other, then maybe we can restore the opposite one. We didn't really see it for males. We saw a decrease in the ability for insulin to bind or interact with the blood-brain barrier. And so this sexual dimorphic pattern also suggests that maybe it hints at why females may be predisposed to Alzheimer's disease. I don't think we have enough evidence to show that females are at a greater risk of developing brain insulin resistance. I don't think those studies have been done, but it would be interesting to see if there is a sex difference in just the development of brain insulin resistance. So this work, as I mentioned, was wrapped up by the MSTAR student, and he was able to present at his won an award and travel scholarship to go and attend the Western Medical Research Conference in Carmel, California in the new year. And then he also gets to present it at AGS next year. So I think he had a fun time participating in this project and really learned quite a bit as a medical student, not having much background in research. So what we can conclude from this section is that intranasal insulin does not impact blood to brain insulin transport into the whole brain or olfactory bulb, which is important therapeutically. If there are high concentrations of brain insulin, it can slow that transport rate. And if the insulin receptor is inhibited, that can also slow the transport rate. It also highlights that there is this feedback mechanism where too much insulin or too little insulin signaling can actually decrease insulin blood-brain barrier transport. And so it begins to link these two processes, blood-brain barrier and brain insulin signaling. We do have some limitations of our study in that we only interrogated the impact of CSF insulin levels on insulin blood-brain barrier transport. So I mentioned we were giving it straight to the ventricle. However, most of the nutrient exchange occurs, is surrounded by this perivascular regulation. And so we don't know what perivascular levels of insulin are or what would happen at the blood-brain barrier. And so we expect if levels of insulin in this perivascular space is high, we would also see a reduction in the amount of insulin transport across the blood-brain barrier. These studies would be hard to do because the space is so small, and so in rodent studies, it might not really be feasible. We also didn't look at the parenchymal regulation. So if we were to inject something directly into the brain parenchyma, such as the areas in the hippocampus, and what would happen to hippocampal blood-brain barrier transport. And so this summer, I really worked hard to generate this new mouse model that has knockdown of this insulin receptor, specifically in the hippocampus. So here we're showing that protein levels of the insulin receptor are knocked down. And what we plan to do in the future is now look at blood-brain barrier transport here and see if it's also impaired. So what are some ways I've presented to you kind of the bad thing that can happen with brain insulin resistance, but what are the ways that we can reverse insulin resistance? And so I'll start first with this project involving exercise, which was started basically the summer of COVID. I had plans to get this NIDDK student that was going to launch this project, and that obviously fell through, but thankfully my team that I had at the time stepped in and was willing to help with these studies. So we all know exercise is beneficial in Alzheimer's disease. It's beneficial in every disease, right? And so we know it can protect our brains, but we're still learning exactly how it's able to do that. So a lot of research in the last couple of years have been focused on these exerkines that are secreted in the blood, BDNF, irisin, lots of these blood factors that are then linked to the improved cognition in Alzheimer's disease. But it's not really known if it's the transport that's enhanced across the blood-brain barrier or if there's something else. And so one thing that I noticed in the literature that hadn't really been looked at is, given this link to insulin and cognition, was could exercise enhance insulin transport into the brain? And could that be a mechanism for the improved cognition? If we get more insulin into the brain, we get improved cognition. And so this graph is to just show that peripheral insulin sensitivity is improved following exercise. I don't think I need to belabor that point. But whether or not exercise could impact brain insulin sensitivity had not really been investigated. So as I mentioned, this team that was with me at the time, including Sarah again, another undergraduate, Noor, a new technician in the lab, Alice, and then actually an undergraduate from Yale that took off a couple of years from her studies to come and do science because she wasn't getting the Zoom interactions that she wanted with her her undergraduate studies, so she took a couple years off to do research. And so she really helped spearhead these studies and get them going. And then Cassidy came in towards the end and helped finish a lot of these exercise studies. So what we did was we exposed mice to this voluntary running wheel. They're able to freely run as much as they want for 24 hours. And then we looked at either blood-brain barrier transport, or we looked at signaling within the brain. And this work's been published, so I'm not going to talk in detail too much. I'm just going to present a couple of bits of the data, and you can go to the paper to read more if you want. And so, like I said, it was published. We were able to be the mouse representation on the cover issue, which was fun. So first we needed to characterize how much exercise these mice were getting. So these are low profile running wheels. They look just like this. They sat in the cage just like this. This is a stock image, but our cages are very similar and looked very similar. So they can run as much as they want between the hours of, we would set up the cages at 4 p.m. the night before, and then we would set them up at 10 a.m. the morning, 10 a.m. one morning, and then we would come in the next day and collect the mice at 10 a.m. the next day. And so the majority of the exercise occurred between the dark cycle, which is 6 p.m. to 6 a.m. because they're nocturnal creatures. So they ran all of this distance and pretty much in that dark cycle. And what you can appreciate is that they are very variable in the amount of exercise that they participated in, we'll say, voluntarily participated in. And what you can also notice is that females ran nearly twofold more than males did. And this has been shown throughout the literature that female mice just run a lot more. Their speed was also a lot faster. So what we showed in this study is that for males at least, insulin transport into the brain was increased nearly twofold. So the slope of this line is nearly twofold greater than the slope of the sedentary line, which were mice exposed to a locked running wheel. So they had the same environment, they just were not able to move their wheel. The females, while we did not see any change in the slope or rate of insulin transport into the brain, we did see that the interactions of insulin at the blood-brain barrier were increased. So this could be maybe the level of the insulin receptor is increased in the brain endothelial cell, which would lead to increased signaling and could still have an effect within the brain. Again, it points to the sexual dimorphic pattern going on between insulin's ability to cross the blood-brain barrier. When we looked at signaling, so we're looking at insulin receptor signaling. Insulin binds the receptor, it gets autophosphorylated, that causes phosphorylation of IRS1, and that causes phosphorylation of AKT. So you can kind of track these signaling events to identify whether brain insulin signaling is impaired. And so females had overall a bit greater level of insulin signaling in the frontal cortex. And then if we look in the hippocampus, we actually saw a significant decrease in the amount of insulin signaling in males and females with exercise. And this was kind of opposite to what we would expect. We would expect that if exercise is improving insulin transport into the brain, we would get enhanced insulin signaling, but that wasn't the case. And it was just localized to the hippocampus. So we're trying to figure out what this data means and if it has anything to do with fasting levels. If the mice are running, they can't eat. And so since food intake and fasting can regulate insulin signaling, we still have to do follow-up studies to identify really what's going on. So to conclude from the exercise work, we showed that females run more than males when they're given access to this voluntary running wheel. This acute voluntary running wheel exercise increased insulin blood-brain barrier transport in the male mice, and it increased regional vascular binding in the male mice with trends in binding for female mice. And so this would still tie into the whole insulin signaling and being able to relay it from this blood-brain barrier. What I didn't show but was presented in the paper is that the run distance did correlate with insulin uptake in certain regions. So that's what we would expect, is if they're running more, they get more insulin uptake into the brain. And so for olfactory bulb, we found this in both males and females. And in the frontal cortex, we found this correlation just for females. And then lastly, we showed that this exercise program actually decreased insulin phospho-AKT, which is a downstream signaler of insulin signaling in the hippocampus. And so all of this work helped to build the basis of an R01 that was just resubmitted this last month and will be reviewed sometime in February or March, in which we want to dive deeper into this finding. So we looked at voluntary running wheel, but as I showed you, that was variable with the amount of exercise the mice were actually getting. So now we want to do more controlled exercise paradigms where we actually put them on a treadmill and they either get this moderate continuous exercise or they get interval exercise, so a bit higher bouts, but they get rests in between. Or we look at a repeated bout effect. So what happens if they exercise for 3 or 4 days in a row? Does the transport rate level out, or does it get further enhanced? And what we're going to look at, what our primary outcomes are, either insulin blood-brain barrier transport or signaling at the blood-brain barrier. And especially, what's going on in the brain? Are we improving brain insulin resistance in this model? Are we improving cognition, which is one of the important clinical outcomes? Are we affecting synaptic plasticity at all? And so hopefully we'll be able to continue with these studies and really learn more about the impact of exercise on the blood-brain barrier and brain insulin resistance. So my last piece of data that I want to talk about, also related to ways to reverse CNS insulin resistance, is using incretin receptor agonists to help improve insulin signaling in the brain. So this is really a joint effort between Bill here at the VA and in the division, and Conrad Talbot, who's at Loma Linda University, but is an expert in brain insulin resistance, especially in the clinical field. He's really driven a lot of this research. And our goal was to look at the ability for these IRAs, these incretin receptor agonists, to actually get into the brain. And this is work that we're just now wrapping up and hoping to get out published soon. So what are incretins? I had to learn about what incretins were when I first started this project because I'm so focused on insulin. But incretins are gut peptides that are basically secreted after food intake, and they help maintain euglycemia. So these are the two main examples of incretins, GLP-1 and GIP, which if you're in the endocrinology world at all, you will know for sure what these are. These receptor agonists are basically substrates that can activate the incretin receptors. And so the two primary incretin receptors are the GLP-1 receptor and the GIP receptor. And so many of these drugs that have been designed target either one or the other, or now more recently, they target both. And so they're called dual incretin receptor agonists. So just like I presented for the effect of insulin in the brain and in the periphery and metabolic field and in cognitive field, GLP-1 action actually follows a similar pattern where it's released in the periphery and then it can get into the brain and have effects on appetite and weight, but it can also have these neuroprotective effects as well. And so it's very similar to what we're seeing with insulin. So it made sense to kind of go after these peptides and potentially try to stimulate their receptors in order to protect in Alzheimer's disease and other neurodegenerative diseases. So this is how a very simplified model of how GLP-1 signals, and you can see how it intersects the insulin signaling to elicit some of the same effects that insulin signaling would. Here is a not quite as simplified model, a more distracting model, I guess, in which you can see GLP-1 binding the cell surface receptor, and then ultimately the effects regarding neuronal development, neuroprotection, and memory formation. And so this is really becoming one of the it's really becoming a greater topic in this field to use these incretins to try to treat Alzheimer's and Parkinson's disease. And there are a lot of clinical trials using a lot of these already FDA-approved drugs for the treatment of type 2 diabetes to now try to target insulin signaling in the brain. And so you can see the amount of publications on these topics has increased just in the last 10 years. So insulin signaling in the brain in terms of memory improvement has really been going on since the late 90s, early 2000s. And this just really started taking off maybe a decade ago. So it's a much hotter topic to use this type of therapy for improving memory. And so this is just a list of all of the incretin receptor agonists that our group has investigated to date for blood-brain barrier transport. And you can see the structure of all of these GLP-1 analogs or IRAs are quite different. Some are really long and bound to albumin. Some are duplicated in their sequence, and some are really short. Exenatide was really one of the first IRAs investigated in this field. Exenin 4 is the peptide that is produced that activates the GLP-1 receptor. And so for the ones that are FDA approved, I've listed their drug name here and how often they're taken, because some are more stable in blood than others, and then when they were FDA approved. And you can see this last one, tirzepatide, was actually just approved this year. And so really we feel like we're right on that edge of looking at the potential therapeutic capability of it in the brain. These ones listed down here are synthetic versions of basically exenatide. This is the base synthetic version, and then every other one is kind of built off of that, activating both incretin receptors. So what has been shown regarding usage of these IRAs in either cognition or development of Alzheimer's disease? And so in some of my literature research, I found that people who used exenatide had a lower risk of developing Alzheimer's disease. So this was probably one of the most interesting studies because it was a retrospective study where they basically looked at insurance data. Maybe you all have problems with that and ways to pick it apart, but coming from the basic science field, this was pretty exciting. And so what they did is they basically looked at exenatide usage for 2 years, from 2007 to 2008 in these people. And then they followed up for 5 years and looked at how many people went on to develop Alzheimer's disease. And what they found is that those that used exenatide a lot and kept trying to get their refills, they had a significant reduction in the level of Alzheimer's disease incidence. And this association was stronger for females, again, potentially leaking this brain insulin resistance and females being more sensitive to it. And the graph on the right is just a graph of some of their table data, where it shows that the most intensive users had the least diagnosis of Alzheimer's disease after that 5-year time point. In the rodent studies, they've shown that these incretin receptor agonist mouse models of Alzheimer's disease. So I'm just giving 2 of the most common examples, exenatide and dulaglutide. They use 2 different mouse models of Alzheimer's disease, but they show that in both of these models, there is impaired insulin signaling. So here, these mice have impaired insulin signaling through this particular protein they're looking at. And here, the gray bars signify that these 80 mouse models have decreased brain insulin signaling. And when they treat with either exenatide or the dulaglutide, they see this reversal of brain insulin resistance. they further went on to show that cognition could actually be improved. So when they gave Exenatide versus saline, they saw improvement in cognition. This is a Morris water maze task. The mouse has to learn where this hidden platform is, and they have to swim over to it and find it. And so for both studies, they showed that in the Alzheimer's disease model, this was impaired, and their IRA treatment could actually improve this cognition. So there was a lot of evidence that there was some sort of action in the brain, but no one had really characterized whether or not it was actually transported across the blood-brain barrier, which is our field, to act directly in the brain, or whether it was maybe just acting at the surface of the blood-brain barrier and then some sort of signal would be relayed on. And this is important when you think in terms of clinical therapeutics, whether your drug is actually reaching the target or whether it's an indirect effect. So we did 2 different types of radioactive transport studies where we looked at the actual transport rate within an hour of all of these IRAs, all 13 that I had listed. And then for a few that did not get in within this hour, did not have significant transport, we looked at a 6-hour uptake and whether these longer lasting IRAs that are more stable in blood just took a lot longer to get in. So what we found is that with the 1 hour, we saw transport of, let's see, 3, 6, 7, 8. We saw transport of 8 of the 13 IRAs, significant transport, getting into the brain. But as I mentioned, these longer lasting ones, in particular semaglutide, liraglutide, and tirzepatide, which are stabilized in blood by binding albumin, they weren't really entering the brain in this time frame. And so we ruled it out. We said, oh no, they must be acting indirectly if they're having a beneficial effect in the brain. But after thinking about it more, we realized that they could be stabilized in blood because they're bound to albumin, and maybe they're just getting into the brain shortly, or they're just getting into the brain similar to how albumin can enter the brain in very low levels. So for this, we did a 6-hour study. So when we compared levels of the radioactive IRA in brain at either 10 minutes or 6 hours, we saw a significant increase. And so this suggests that after 6 hours, eventually these peptides, these drugs, can enter the brain and still have some sort of clinical therapeutic effect. So giving it systemically, giving these drugs systemically is fine if a person has type 2 diabetes or difficulties in regulating blood glucose, since that's what they were primarily designed to do. They primarily regulate glucose levels. But if you're giving that to a healthy person that doesn't have hyperglycemia, then you have an increased risk for giving them hypoglycemia if you give these drugs, because it's going to take all of that glucose out of their blood. So we've always been interested in this dual administration, systemic and intranasal. And so we wanted to see if we could deliver these incretin receptor agonists through the intranasal method and still get it to the brain as an alternative way to deliver it in the clinic, just like intranasal insulin. So what we found is that on the left, you can see the amount reaching the brain after given intravenously, so systemically, and on the right, you can see the intranasal levels. So they are still significantly decreased intranasally, but we're still getting some in. And so the next steps are going to be needed to define whether the amount that's getting in is still therapeutically beneficial, if the amount reaching the brain can still activate the signaling pathways it needs to. So for my final conclusions from this section, we were able to show that albiglutide and dulaglutide, which are just single incretin receptor agonists, they have the fastest blood-brain barrier transport rate. We showed that the longer lasting drugs that did not show transport within an hour did actually exhibit uptake over this 6-hour time frame, suggesting that they can eventually get in. And so for treating CNS disorders with brain insulin resistance, we want to limit the side effects, particularly on the blood glucose levels. And so we showed that we were able to give it intranasally and it can distribute throughout the brain and therefore may still be therapeutic. But it is less effective than blood-brain barrier transport. And so to finally conclude, this IRA off-label use for CNS disorders with brain insulin resistance may actually be beneficial in reversing this brain insulin resistance and improving cognition. So a lot of follow-up studies need to be done to identify how active they are once they're in the brain. We've shown that they can get into the brain. So with that, I'd like to thank my group, in particular, all of the students that worked on this project, my current students, the greater VA blood-brain barrier group we had by Bill Banks, and Shelley has really provided a lot of input on a lot of these projects. Kim worked with a lot of the incretin receptor agonist studies. May Reed's been really helpful in a lot of the vascular studies. And so with that, I'd like to draw you all to any questions or any discussion. Shelley, go ahead. Yeah.
Sorry, can you hear me?
I can hear you now.
Okay, great. So first, great talk. And I actually have two questions for you. So in the beginning, you mentioned, ways to determine whether people are CNS insulin resistant. And I'm wondering if there are any good clinical diagnostic tests for that yet. What's the best way to determine that in people who are still alive?
Yeah, so that is a great question and something that I wish I had had more time to get into that we covered in a recent review that we just put out. But you're dead on. That is the problem, is that there is currently not a good clinical test for brain insulin resistance. A lot of it is based on CSF draw, so a lumbar puncture and looking at the blood to serum ratio to see if that's off regarding insulin levels. We can't do a HOMA IR, or we can't do a glucose clamp in a lot of these patients to look at brain signaling. That's all peripherally mediated. I will say, excitingly, there is a new insulin tracer. I think it's a PET tracer. And so potentially down the road, we could actually look at blood-brain barrier transport of this insulin PET tracer in humans to see if that's at all changed over the course of disease. The other test is just standard PET signaling that a group out of Germany has really spearheaded in looking at insulin activation of neuron firing in the brain. And so they kind of use that as an indirect way to look at neuronal insulin signaling. But it's still, it's hard to do unless you have that postmortem tissue.
Yeah, that's really cool about the insulin PET ligand. Second question is, what do we know about brain endothelial cell insulin resistance? So, do we know anything about whether their ability to, signal in response to insulin is altered with aging or Alzheimer's disease?
Yeah, so another great question. So a group out of Canada, Frederick Cologne's group, just published earlier this year that in microvessels isolated from humans and mouse, all Alzheimer's disease mouse models, saw disruptions in this insulin receptor signaling pathway. And I think they actually saw decreases in the level of the actual insulin receptor, and it was one particular isoform of the insulin receptor. There are two isoforms, with one being primarily predominant in the brain. And so they were able to show that at the blood-brain barrier, this process seems like it's impaired. But that's hopefully what this grant will start getting at, is looking at blood-brain barrier signaling and how that relates to brain insulin signaling, because that hasn't really been linked yet. We always think of just a substrate getting across the blood-brain barrier and getting into the brain, and that's the main goal. But the blood-brain barrier, that endothelial cell itself, is a signaling interface, and we want to know how that signaling relates to the brain. Great question.
Very cool.
There was one question in the chat. I don't know if you're able to see the chat.
Yes, now I
from
can.
you.
Okay, so Sophia asked, do you think reversal brain insulin resistance is the key to weight loss produced by incretin receptor agonists? Great question. I will say my knowledge on the metabolic side is more limited. I would refer the question to Bill, but I will take a stab at it. I think that is one of the goals of a lot of these drug companies, is to look at it more in its metabolic role within the brain rather than its cognitive role. But of course, if a drug company can create this drug that affects two huge diseases in our population, both metabolic syndrome and neurodegenerative disease, then that's going to be a big hit for them. So I think while most of them are still looking at the metabolic function of these incretins within the brain, some, like Novo Nordisk in particular, is now starting to creep into that Alzheimer's disease, neurodegenerative disease field. And thank you, Ton, for your comment about working with the trainees. I really do enjoy that. And I know Cynthia kind of leads it, and I look forward to a summer student every year if I can take them on.
Yes, thank you, Elizabeth. You're an awesome mentor.
Thanks.
Hey, this is Angela. So I do the human equivalent of this stuff. To get to Sophia's question, I believe they have multiple effects on the gut. So semaglutide in particular, the huge side effect that we have to counsel people in our consent form is weight loss and nausea. And I don't think it's, I mean, they certainly may have effects on the brain, hypothalamus and all that, but I think it's gut specific, you know, it binds to this and that and the other thing and affects some gut hormones. And it's immediate, like the first time they take a dose. So I think this is gut related.
Yeah, it would be interesting to see what the intranasal dosing would do if it had that same gut effect. But
yeah, if you could bypass that a little bit, and maybe you wouldn't get the weight loss. What we don't care about that for Alzheimer's, you know. So you could have a separate, you know, formulation. Of course, as you all may know, in your world, Elizabeth, you know, intranasal, I mean, you have your own problems with trying to get intranasal dosing. I think you have to sedate your patient.
yeah.
In our world, you don't have to sedate the patient, but intranasal dosing is tricky. And the intranasal insulin phase 3 study was a failure. And we think it's because we didn't get target engagement. In other words, the atomizer didn't get up into the cavity because our head's too big. So,
exactly.
So it's not, you know, it's an engineering problem, which I'm, as a scientist, I'm like, oh, come on, like, really?
Yeah. All right, Bill, did you have a question? Thanks,
anyway.
Angela.
Yeah, I was just going to address about perhaps it's CNS mediated, and I just want to point out that with insulin, the insulin, and this was off, but if you give insulin into the brain, then in the periphery, glucose levels go up, insulin levels go down, and feeding usually goes down as well. The effects are mild, but we suggested, based on that about 20 years ago, that maybe the CNS insulin is acting as a counter hormone, sort of to the peripheral effects, sort of closing a feedback loop. But of course, your work has taken that idea, advanced it way beyond what we suggested 20 years ago.
Thanks, Bill. So we're right at time. If no one else has any other questions, I'll let you go for the remainder of your year. Enjoy your holidays. Have a great new year.
Thank you so much.
We'll see you next time.